after the hour in Woods Hole. So to have the high school science fair winners from both Falmouth High School and Falmouth Academy with us today, and that's always a great treat to see students venture off into some of their first inquiries into science and do such an outstanding job. And because the Oceanographic Institution always honors the top winner at both schools with a scholarship award, I'd like to introduce John Farrington, the Vice President for Education, who is the one who always gives out the checks, and so he could offer a few words of wisdom to the group.
Well, thanks, Judy. I'm not so sure about the words of wisdom, but I can give you some words of enthusiasm, and that is that I always consider it a great honor to represent the institution in participating in the science fair at Falmouth Academy and at Falmouth High School and the public school. And I enjoy very much each year looking at the different presentations and the posters, and I'm very pleased that you've all joined us here today. And I look forward to these particular presentations that we're going to hear from these wonderful young scholars who are carrying on in the tradition of science in the Falmouth community that's been a long tradition. And I think that it's fair to say, and I hope our superintendent will agree with me, that the partnership between the scientific institutions in Woods Hole and the Falmouth schools and the Falmouth Academy has been one of these wonderful community efforts. And I have certainly enjoyed, ever since coming here in the summer of 1971 as a postdoc, participating in these programs. So thanks very much for coming, and it's an honor to be here.
Thanks, John. Now, for those of you who are regulars, we usually have cookies out, and some come just for the cookies. I know they're looking for them at the back entrance. But because it's the high school and academy science winners, we have special desserts tonight after the talks. So you can save your appetite for the end of the presentation. And to kind of introduce our science in Falmouth, the commitment of the Falmouth community to science excellence, Tracy Crago, Sherry DeRosa, and Andrea Thurwald put together this slideshow, which will introduce, science in the community. It should be launching soon.
I've got to keep on keeping on. I just want to be beside you, want to do the best I can, even though you've made it plain you don't believe in love. I can't let you go. I can't walk away. Come on, baby, can't you see all the need in me? Like the ocean speaks to the naked man. The rhythm of your heart is like a sonic blues sweet. How you hold my memory, believe me, baby. In this brilliant world, you're the best. You're like porcelain charm, a perfume smile, a teenage blush. You're like champagne roses. Come on, baby, can't you see all the need in me? Like the ocean speaks to the naked man. I've got to keep on keeping on. How can I live without you? Got to make you understand. Each species has a mate, and fate says you'll be mine. I won't let you down. Come on, baby, can't you see all the need in me? Like the ocean speaks to the naked man. The rhythm of your heart is like
some of you recognized yourselves in there, either as judges or participants. But I hope the students enjoy the science fairs as much as the judges do, but the judges enjoy it an awful lot. So the 4 presenters today are the recipients of a lot of hard work and a lot of high honors for their presentations. And the girls have decided, we have all women today, and the women have decided that they are going to present in which order. And the first one is going to be Sarah Shepard. And Sarah is a junior at Falmouth High School. She earned a first place award for her project, The Effects of Potential Endocrine Disruptors on Daphnia magna. Sarah?
Good afternoon. Today I came to talk to you about my science project, Effects of Potential Endocrine Disruptors on Daphnia magna. Endocrine disruptors are compounds that mimic natural hormones and can disrupt an organism's endocrine system. For example, endocrine disruptors such as DDT have been shown to disrupt the sex ratio in alligators in places such as a lake in Florida. Potential endocrine disruptors can naturally occur in plant products and animal waste. The objective of my study was to determine the effect of several endocrine disruptors on the life history of a Daphnia magna. The test subjects in my experiment were exposed to low levels of estrogen, which in this case I used a mare urine, a phytoestrogen, which I used soybean, and a testosterone mimicking substance, which I used white pine pulp. The test subjects of the experiment were Daphnia magna, which are common aquatic crustaceans, and they were ideal for my experiment because they are an important basis to the food chain and they also reproduce asexually. This is a brief summary of the materials I used, and for the wood pulp, I made a leachate using wood pine chips cut up about 1 centimeter by 1 centimeter. And I took the sterols from that white pine chip leachate, and in the presence of microorganisms, they were converted to a testosterone mimicking substance. With the pregnant mare urine, I made a solution, and the 17-beta estradiol in the presence of microorganisms was converted to an original estrogen molecule. And for the soybeans, I ground the soybeans up, and the genistin in the presence of microorganisms produced a phytoestrogen. The test setup was 6 solutions and 2 concentrates per solution, 7 replicates per solution, and 2 control solutions, and 1 soybean solution that was filtered through a liter of silica sand. This was to represent like a soil column, and when a soybean solution or any phytoestrogen would leach through the ground. There was a single daphnin placed in each of the beakers and exposed to the test substance for 21 days. And each day after the young were released, which was around day 8, they were counted and removed. At the end of the study, the lengths of surviving daphnids were measured using a dissecting scope with a calibrated micrometer. This is the length data, and you can see that as with the orange, they are the urine solutions, and the soybeans are the royal blue ones. And as the estrogen as the amount of estrogen within the solution becomes greater, the average length becomes smaller. This is the soybean solution through the soil, and compared to the previous graph, you can see that the sand column reduced the effect the estrogen had on the Daphnia compared to the control. And finally, of the length is the testosterone mimicking substance, and there seems to be no real effect that the testosterone had on the average length of the Daphnia. In these pictures, you can compare with the two soybean solutions compared to the control, that the average length of the animals were smaller, and the soybean, the greater percent of the soybean looks unhealthy compared to the control at the top there. These graphs are for the molting, and molting is important to crustaceans because that's how they grow. So you can see that, again, as with the lynx, that the greater amount of estrogen within the solution cause less molting than the control. And with the silica sand testing, again, the sand reduced the effect that the estrogen had on the Daphnia. And finally, with the testosterone mimicking substance, again, as with the lynx, there seems to be no real effect that the testosterone had on the average molting. These graphs show the average young produced by the animals, and there is a significant decrease in the amount of young produced compared to the control, with both the high percent of the urine and the soybean. So the estrogen greatly affected how the young were produced, or how many young were produced. And the silica sand column reduced the effect that the estrogen had on the amount of young produced. In the testosterone, as with the molting and with the lynx, there's no effect whatsoever on the amount of young produced. These graphs just show each replicate within each solution, and you can see that each replicate within each graph, the lines are about the same. So there was each replicate produced about the same amount of young, but with the control compared to the high mare urine, there was a significant decrease in the young over the period of the 21 days. This is the soybean, and it's the same setup where the amount of young are on the y-axis and the days are on the x-axis. And you can, again, as with the urine, there is a significant decrease from the 0.01% soybean to the control. And with the soil treatments, again, it's the same setup, and there's not a significant decrease, which the other average graphs had shown before, between the control and the soybean, which meant that the soil reduced the effect, but this graph shows that there is more variability between each replicate and the soybean. Finally, with the wood pulp treatments, you can see that the graphs are about the same, and there is not as much variability between each replicate, which shows that there is not much of an effect that the testosterone had on the amount of young produced between the replicates. In conclusion, the estrogen affected the growth, the reproduction, and the molting of the Daphnia. The soil reduced the effects the estrogen had on the Daphnids, and the testosterone mimicking substance did not seem to affect the growth, the reproduction, and the molting. I would conclude that any material containing an estrogen or an estrogen mimicking substance that directly contaminated an aquatic system can have a negative effect on the growth, the reproduction, and the molting. However, if a similar material entered the system through leaching through sandy soil, it would have reduced the effects on the daphnids. And I would just like to thank Arthur Putt and Leslie Whitney. That's it.
Our next speaker is Phoebe Poole, and Phoebe is a senior at Falmouth High School, and she is just accepted to go to Wellesley next year. So she'll be taking her Falmouth science experience beyond the boundaries of Falmouth. And the title of her project is Host Range of the Endoparasitic Dinoflagellate Amoebophyra. Get this one off.
Good afternoon. My name is Phoebe Poole. I go to Falmouth High School, and my project is entitled Host Range of the Endoparasitic Dinoflagellate Amoebophyra, which was isolated from Alexandrium tamarense. To begin, I'm going to introduce the host and parasite that I used in my project. And as the host is a red tide species, which causes algal blooms, I'm going to talk briefly about those two. To begin, the primary host in my experiment, Alexandrium tamarense, is a dinoflagellite, which is a kind of algae. The life cycle of the organism is made up of sexual and asexual steps, as well as a hard-bodied cyst which settles to the bottom of oceans and lakes. And it is that cyst that is responsible for blooms, as favorable temperatures can cause the cysts to break out and form into the regular motile form of the organism. And then those cysts can be forced back into the cyst stage by overgrazing by parasites or unfavorable temperatures. Red tides are caused by a toxic or non-toxic algal species that grows and accumulates in a certain area. Toxic species can kill marine fauna by releasing toxins into the water, or they can damage the gills and internal organs of fish. Toxic dinoflagellates in the genus Alexandrium, which was the primary host in my experiment, produce paralytic shellfish poisoning toxins that can accumulate in fish and shellfish and then cause harm to humans. However, non-toxic blooms can also be harmful in that they can deplete the oxygen in the water or block out light needed by bottom plants. The parasite in my experiment is called Amoebophyra, and it's also a dinoflagellate like the host. However, the life cycle is very different. It begins with a small dinospore, which is free-living in the water, which then enters into the host and develops. And then once it breaks out of the host in the vermiform, it can break up into dinospores and go on to infect other hosts. As Amoebophyra, when it enters into the host is castrated and cannot reproduce, and then when the parasite emerges out of the host, it destroys it. And because they must kill their host to complete their life cycle, and because of high infection levels that have been observed in some hosts, it has been suggested that the parasite could be used as a biological control for red tide blooms. This shows more clearly the parasite. The first figure shows the parasite attached to the host nucleus. The second shows the parasite once it has entered into the host nucleus. In 3, you can see that the parasite has begun to grow, and there are several nuclei that are visible. And then in 4, the nuclei have developed even more, and there are many of them arranged in a spiral. Number 5 shows a late-stage infection of the parasite in the host, which is a characteristic beehive shape. And then 6 is the vermiform, which is a free-living form in the water outside of the host. So in order to evaluate the use of the parasite as a biological control, I wanted to answer the question, what is the host range of Amibophoria when isolated from Alexandrium tamarense, and does the parasite have a host preference? I hypothesized that the parasite would have a narrow host range, as recent experimental work has been unable to infect dinoflagellate species other than the primary host. And this is due to recent work that has been done. Up till a couple years ago, Amibophoria had been observed in about 30 different species representing more than 20 genera of dinoflagellates in locations anywhere from the Mediterranean Sea to the Atlantic Ocean. And when scientists tried in the lab to infect other hosts other than the primary host, that was unsuccessful. In this presentation, I am not going to go into the details of my procedure as it's quite extensive, but the details are included on my board, which will be displayed after. So to begin my experiment, I cultured my primary host and the 6 other hosts that I will be using in my experiment, which are listed here. The first is the primary host, Alexandrium tamarense, which comes from Perch Pond in Eastham. Then there are 2 more Alexandrium species. Alexandrium fundiense comes from the Gulf of Maine. Karenia brevis, the one at the bottom, comes from Florida. And you can see that the hosts represented here come from quite a range of places. The Scripsiella trichoidea is from South Africa. So these hosts are all quite different, and some of them are different in shape also. So to begin some cultures of parasite that I would use throughout my experiment, I counted a host and parasite culture after I'd filtered the parasite to collect the small infective dinospores, and then I combined the uninfected host and parasite dinospores in a known ratio. In my first study, I began infections of the 7 different hosts, including the primary host. After 5 days, I counted the infections, and an infected cell was one that had a late-stage infection in it. And under a fluorescent microscope, the parasite fluoresces green and the host fluoresces red. And this shows a late-stage infection where the parasite is quite large and ready to emerge from the host. In my second study, I infected the same 7 hosts, yet this time the dinospores that I used to infect the hosts came from a single isolation that I had done, where I isolated a single infected Alexandrium tamarense cell, and from there I scaled that up and made clones, and then filtered those to get the dinospores. Again, I combined the hosts and parasite, and then on days 4, 5, and 6, I sampled, fixed, and counted the infected cultures. From my first study, I found that all 3 Alexandrium species were infected, which are these 3 here. This is the primary host, and I was also able to infect Heterocapsa. In the second study, again, I was able to infect the 3 Alexandrium species, including the primary host, again, Heterocapsa, and I was also able to infect the Scripsiella. From my first study, I concluded that the parasite Amibophoria had a much broader host range than hypothesized when it was isolated from Alexandrium tamarense, and that there was a significant difference between the infection rates of the different Alexandrium species, meaning that in my first study, you can see that although all 3 Alexandrium species were infected, it was actually the primary host that was infected the least, whereas the Alexandrium from South Africa was infected more successfully than the primary host from Eastham. From my second study, I concluded again that Amibophoria has a much broader host range than I had hypothesized. Again, there was the difference between the infections of the Alexandrium species. And also, based on this study, I said that the parasite is not able to infect athecate dinoflagellates as its primary host is a thecated species. Meaning that in the second study, you can see that all 5 hosts that were infected, those 5 have cellulose plates in their body wall, which means they are thecated, whereas the 2 that I was unable to infect are athecates and do not have the cellulose plates. As can be seen by the conclusions, the parasite would not be a successful biological control as it is not specific enough to its host. Yet though it is not a viable biological control, the parasite will actually be more successful as a non-specific species, meaning that in an ocean or a lake, if the parasite's primary host is not available because of environmental reasons, then the parasite will still be able to survive by infecting other hosts. Also, this study is helpful in understanding the little-known host-parasite relationship and also adds to the knowledge about phytoplankton and harmful algal bloom species, which are of major concern as of late with the algal blooms that have been occurring. For future study, it would be even more helpful to evaluate the host preference of the parasite to further look into its use as a biological control. And that could be tested by mixing 2 hosts together in equal ratios and seeing which the parasite would rather infect. And I wanted to thank Dr. Mario Senko, who I worked with here in the biology department, my teacher, Miss Brothers, at Falmouth High School, and my boss, Bruce Wooden, for allowing me to go work in another lab. And that's it. Any questions? Yes. Okay. That might be true. I mean, the hosts that I was growing were all grown, like in the lab. They were cultured in the lab in a standard solution, which was an F/2 media, which was just filtered seawater with minerals in it. So I'm not sure if that would make a difference.
Will be available after. Our third speaker this evening is Megan Starr. And Megan is a sophomore at Falmouth Academy. And today she'll be talking about her project, Ozone Depletion and White Pines.
Hi, I'm Megan Starr, and actually there's a slight mix-up. My project's about ozone pollution in white pines, which is kind of different than ozone depletion, and I'll tell you about it. And I compared the ozone pollution, I figured out the effect of ozone pollution on white pines by measuring 3 variables for white pines, which were the average needle length, tip necrosis length, and chlorotic model of different white pine needles, and compared those variables to the history of ozone pollution in each area. Ozone in the stratosphere, which is the second layer of our atmosphere, is actually vital for life on Earth. It absorbs ultraviolet radiation from the sun, specifically UVB rays, and they can cause skin cancer, eye cataracts, and wrinkles. Ozone pollution, however, occurs when ozone is present in the troposphere, which is the level of our atmosphere that we live in on Earth, and it's extremely damaging to tissue living things. It's formed when volatile organic compounds and nitrogen oxides react with the sunlight. And it's the most widespread pollutant in Europe and North America. And it's responsible for reducing lung function and capacity for exercise, and it also aggravates or increases susceptibility to asthma, bronchitis, pneumonia, you name it, basically a bunch of lung diseases. And it also can cause permanent lung damage, even if you're only exposed to it for short periods of time. It also affects plant life. It can reduce crop yields, destroy forests and vegetation, and it does this because it reduces the ability of plants to photosynthesize. And the trees that I used in my project are white pines, and this is a cross-section of a white pine fascicle, which is a bundle of 5 needles that is characteristic of a white pine. And they're the largest of the northeastern conifers. They like well-drained sandy soils, like basically around here. And the needles are around 2.5 to 5 inches long. And you can see they come in this formation. The 2 inner sides are the stomatal sides, and the outside is a non-stomatal side. And one of the variables that I was measuring, chlorotic model, occurs when ozone enters into the stomatas of the pine needles and it kills the mesophyll tissue, which is where photosynthesis takes place. And it causes this yellow spotting all around the stomatas of the needle, which you can see in the graphic on the left. And that's a lot different than chlorotic fleck, which is on the right, that you have to distinguish from, because chlorotic fleck is caused by abiotic factors like salt damage or different chemicals getting on the needles. Tip necrosis, which was another one of the variables measured, is just the death of the tip of the needle. And it's usually an equal length for all of the needles in a fascicle. The purpose of my project was to measure the amount of ozone damage to the white pines in different areas and compare the amount of damage to the history of ozone pollution in each area. And it was hypothesized that white pine trees that are in areas with elevated levels of ozone would show greater ozone damage by having a shorter needle length, more tip necrosis, and more of chlorotic model. My methods, I had 12 areas around New England that I collected needles from, and these are the locations. I got 3 trees from each of the locations, and I sampled 10 needles randomly from the tree. And then I measured the total length and the length of tip necrosis using just a millimeter ruler. And then to determine the chlorotic model of a needle, I used a clear graph paper transparency, and I put it over the needle and looked at it under a microscope, and I could count the total number of squares the needle took up and also the total number of squares the chlorotic model took up to calculate a percentage of the needle covered. And once the needle is 40% covered, then it's basically useless. It can't photosynthesize anymore. And then to determine the history of ozone pollution for each area, in the Clean Air Act in 1990, the EPA, which is the Environmental Protection Agency, set guidelines to limit the production of ground level ozone, and they set a health standard at 120 parts per billion of ozone in the air for a 1-hour average. And the standards were tightened in 1997. So the health standard now is 85 parts per billion of ozone in the air. These are the ozone monitoring stations in northern New England. The pink dots are the locations that I sampled trees from, and the line connects them to their corresponding ozone monitor that I looked at. And this is southern New England, where I also collected trees. For each of the 8 sites, there were 8 sites that I found corresponding ozone monitors, and I went through the archives of the EPA for the past 3 years for the ozone monitoring sites, and I could find the number of days that each area violated that each area's air violated the health standard, and I could also find the parts per billion that violated it every day, that it was in exceedance, and so I could calculate the total parts per billion that it had exceeded the standard. This is my first graph. On the y-axis is the average needle length, and on the x-axis it has the locations in order of the number of days that it violated the health standard. So on the right, Fairfield had the most violations. And the black line down the middle is the best fit line. This is the second variable, which is the average tip necrosis length, and along the x-axis, again, are the locations in order of number of days that they were in violation of the standard, and there's a best fit line for that too. This is the third variable, which was percent of the needle covered in chlorotic model as compared to the location in order of number of days that it violated the standard. This is a table of the averages for each location and also the ozone data. so for the analysis, the best fit lines on my graphs, they sloped in the hypothesized directions. However, the R-squared values were very low and didn't support a correlation. For the three areas with the least ozone pollution, which were Middlebury, Vermont, Lincoln, and Chatham, their needles had no chlorotic model, an average tip necrosis length of less than a millimeter, and a healthy needle length of over 86 millimeters. And an explanation for this was, despite having a high regional level of ozone, specific areas within a region, such as in the middle of a forest or by the side of a parking lot, where the emissions, the VOCs and nitrogen oxides that cause ozone are being produced, could have a lot more ozone pollution than an area within the same region, but in a place like a forest where the air is probably cleaner. An example that supported this was I took needles from the Falmouth Academy parking lot. The average length was 85 millimeters, tip necrosis was 6.1 millimeters, average percent of the needle was 12%. And then less than a quarter mile away at an FA soccer field, the average length of needles was 100 millimeters. Average tip necrosis, there wasn't any, and the average percent of the needle covered on chlorotic model is around 3.8%. And this is also shown in the needles from Fairfield, Mass, because they were taken from a very rural, woody section, and the ozone measurements were taken near the busy city of Amherst, and so they had a very healthy length, not a lot of tip necrosis and no chlorotic model, even though the ozone ratings for that level were higher than a lot of them. And just one of the reasons that explains why we're having such an ozone pollution problem right now, especially, for example, Truro, which has one of the highest ozone pollution levels in Massachusetts, is they call us the tailpipe of the Midwest. All of the urban rural centers that are producing all these emissions, they float up into the atmosphere, and wind pushes them up the coast, and they basically run right into Cape Cod. And the amount of ozone in the air spikes about 150 miles away from the source, because it takes that long for the sunlight to get to the emissions and react with them. So we're getting the brunt of a lot of the emissions from places that are producing them. During droughts, the stomatas of pine needles close, and that can prevent ozone from entering into the needle and reaching the mesophyll tissue. And also, as I talked about before, regional levels of ozone may not accurately represent the exact area where the needles were from, and many other factors can affect the health of trees, like nutrients there and ages. For improvement or to go on in the future, an experiment could focus on one region and take needles from different places, such as a parking lot or the forest, to compare those variables. And then I could also use a larger sampling group of trees and one age to cut down on or average out the variables. And there's some scientists that are doing work on this out in the West, but they're working with Jeffrey and Ponderosa pines that I could look into. And so in summary, ozone pollution is starting to become a really big problem in North America, but not a lot of people are noticing yet because it just hasn't become an issue. And all of these products are reacting to form huge amounts of ozone, which are severely affecting the health of humans, animals, and plants. The more we learn about ozone and its negative effects, then the sooner we realize that they need to tighten emission standards. And these are just in some articles from the Cape Cod Times about ozone on Cape Cod. This is the Miron Electrical plant that a lot of you guys know, and they're trying to delay having to comply with emission standards for 2 more years. And the results of just a delay, not what they're normally giving off, for an additional 15.75 million pounds of nitrogen oxides, and they estimate it's going to cause 87 premature deaths, 940 emergency room visits, just a lot of bad things. And this is just from one plant, a 2-year delay in emissions, not what they're already giving off. And this is more recently how all of our national seashore beaches are flunking the National Health Standard. And I would like to thank my teachers.
Our last speaker this afternoon is Stephanie Pomeran. And Stephanie is a freshman at Falmouth Academy. She also captured a first place award, and the title of her project is The Effect of High and Low Sound Frequencies on Artemia Selena, modeling the effect of the proposal of Nantucket Sound Wind Farm on the surrounding aquatic environment.
The purpose of my project was to test 2 different frequencies on Artemia selena, which are also known as brine shrimp. This was to model the effect of the Nantucket Sound Wind Farm on the surrounding environment. I got this idea because right now the idea of whether we should put the Nantucket Sound Wind Farm in or not is under extensive debate, and there isn't very much research on the aquatic life and what the effect of the wind farm would be on it. Previous studies have said that 500 hertz and 30 hertz may affect fish, and prolonged construction may affect fish nurseries and may alarm fish. But once the farm is operating, fish will come in abundance around the farm. I hypothesized that the 30 hertz would affect the brine shrimp more than the 500 hertz, and that this would be expressed by them moving quickly away from the source of the sound and then returning slowly, building up tolerance for it. What I did was I had a tank that was 2 feet by 1 foot long, and I put salt water into it. I did 2 replicates for the 30 hertz and the 500 hertz. And I got the 30 hertz and the 500 hertz because it is the expected range, high and low, for the Nantucket Sound Wind Farm. For each replicate, I used 50 brine shrimp, and I put them into the middle of the grid, and then they went to the sides. Then for 30 minutes, I counted how many brine shrimp were in each grid section, and there's 10 grid sections, and the first one is with the speaker in it. Here, the light blue section at the top is grid region 1, which is the one with the speaker in it. And the light purple is the grid region farthest away from the speaker. And on the x-axis is the start and end time for each trial for the 30 hertz, trial 1 and trial 2, and then for the 500 hertz, trial 1 and trial 2, and then the control. And then it's 1 through 50 on the y-axis, which are the number of brine shrimp in each. So at the beginning of the first trial for 30 hertz, there was about an equal amount on the side with the speaker and then on the other end of the tank, and only a couple in between. At the end of 30 minutes, most of the brine shrimp had migrated to the end of the tank with the speaker, other than a couple last remaining ones. And those ones may be due to either there was a light in the other end of the room, and so they may have been attracted to the light because brine shrimp are attracted to light, and so they may have been attracted to the light over the sound. Or it may be there might be a heightened noise in that end of the tank because the frequencies are bouncing off from the speaker on the other end of the tank. For trial 2, basically the exact same thing happened. And then for 500 hertz, surprisingly the same thing happened again. And it was interesting that for all 4 trials, almost the same number of brine shrimp ended in each trial in the grid with the speaker. And then for the control, basically the opposite happened, and more of the brine shrimp went to the other end of the tank without the speaker, even though the speaker was in it. So that may be due to the fact that there was the light in the room. My hypothesis was partially supported because I thought that the 30 hertz would affect the brine shrimp more than the 500 hertz. And this was true, but not by a significantly large amount. The study suggests that 30 hertz and 500 hertz both affect brine shrimp. And previous studies say that future effects may be vibrations will affect communication and affect brine shrimp and fish in sensing predators coming and escaping the predators. So for a future study, I would like to test the effects of the sound and introduce a predator into the situation. So if I have a setup with 30 hertz and 500 hertz playing simultaneously with a control, and if more brine shrimp get eaten from a predator with the speaker playing, then maybe this could be a bad thing and they can't sense the predators coming and so get eaten more. So as a result, this may be a good thing because the Nantucket Sound Wind Farm could produce a nursery-like area because it's already going to be shallower and the brine shrimp will be attracted to the sound. Or it could be a bad thing if the predators are able to eat their prey easier and fishermen may completely wipe out that supply.
How is it that they sense the sound?
For the predators, like when the predators are coming, how they sense them. I don't know, maybe that's how they sense the predators coming, is some way that the frequencies may distract that. I don't know what they use, but something that will cause that to be not work, and so they can't sense the predators coming and they can't escape the predators then.
All of the students have their posters here, so I'd like to come up to the left, the dessert down here at the table. But I'd also like to say that, the students are influenced by a wonderful group of science teachers in this community as well. Don't put all on your own, and don't strictly serve this as a judgment for the measures they have. They have a wonderful group of science teachers, both at Falmouth Academy and Falmouth High School. But I'd like to recognize especially one, Bruce Brother, who is the recipient of that Presidential Award for Science Teaching Excellence. So, Chris, that's a nice subject. And he recently spent a week in Washington with other teachers from around the country and U.S. presidents.
Can I say,
Yes,
okay, like 7 or 8 years ago, I had a fellowship at Hui with John and Judy, and it was a fantastic opportunity to research shellfish diseases, actually. But I worked there for 3 summers, and it was the greatest opportunity, and known by all states, highly leading scientists from the scientific community today, so I would definitely recommend instruction in that role.
So, you all can meet the students one-on-one, and their teachers and mentors, and thank you for coming. Next week's presentation is also an early afternoon, a 4 o'clock presentation, and it'll be the television meteorologist Mike Wankum, The World of Weather Wisdom. So hopefully you can join us for that. Thank you.